Hello, everyone. My name is Ron Vale from Janelia Research Campus, and I'd like to welcome everyone to Life Science Across the Globe. We're now in our second round of talks, and it's great to return to the continent of Africa and our sister institute, the Institute of Infectious Disease and Molecular Medicine. So right now I'd like to introduce Valerie Mizrahi, who is the director of this institute, and she's also faculty in the Department of Pathology at the University of Cape Town. Also in the past, Val was connected to HHMI as a senior international research scholar. So Val is a very accomplished scientist, and her work has focused on Mycobacterium tuberculosis, studying this pathogen from basic cell biology to disease mechanisms to drug discovery. She received the L'Oreal UNESCO Award for Women in Science and the gold medal from the South African Society of Biochemistry and Molecular Biology, and the platinum medal from South African Medical Research Council for lifetime achievements. So Val also gave a great talk for Life Science Across the Globe in the first round, and I would encourage you to watch that talk as well. So Val, thank you, and I'll turn things over to you so that you can introduce today's speakers.
Thanks, Ron, so much for the introduction. It's an absolute pleasure for me today to be introducing two speakers from the IDM at the University of Cape Town. Our first speaker, who will be giving the science talk, is Tom Scriba. Tom is professor in the Department of Pathology and the Deputy Director for Immunology at the South African Tuberculosis Vaccine Initiative, SATVI, a research center based in the IDM and a global leader in TB vaccine research. Tom trained in biological sciences at Stellenbosch University here in Cape Town before going across to the University of Oxford, where he did a DPhil in T-cell immunology. He then came back to South Africa to join UCT, where he worked in SATVI initially as a postdoc and then made a very rapid rise to his current position as deputy director. Tom's research focuses on the immunology and the immunopathogenesis of tuberculosis, TB vaccine design, and biomarker development. He has been a co-investigator on more than 24 clinical vaccine trials and is very well published in this field. His research is funded by grants from the Bill and Melinda Gates Foundation, the NIH, the European Union, as well as the South African Medical Research Council. In recognition for his outstanding achievements as a young investigator in research, Tom received the silver medal from the South African Medical Research Council, and today he'll be telling us about vaccination against tuberculosis, old dog and new tricks.
Well, good morning, good afternoon, and good evening to you all, and thank you very much for that kind introduction and for the invitation to tell you about something that I'm very passionate about. Yeah, so I'm going to tell you a vignette that cuts across what's been happening in the TB vaccine development space over a number of years now. I'll tell you a little bit about a very old vaccine, the old dog, and some new tricks that look a little bit more promising. Of course, as is tradition now, we have to put this all into context. And the current context is, of course, coronavirus. So you will see on the right-hand side at the top that the number of deaths due to COVID-19 has this month surpassed those for tuberculosis. Clearly, the efforts that are being put into COVID-19 and the attention it's getting are appropriate. But I would say that given the number of people that are suffering and dying from tuberculosis, TB is definitely incredibly underappreciated and does not receive the attention it deserves. On the left-hand side are the number of media mentions. This was accessed on the 17th of September, and you can see that the massive orange block, 2.1 million mentions in the media for coronavirus, whereas TB is hidden away by in that little block pointed out by the yellow arrow. So why do I say this? Why is TB underreported? Well, TB has killed many people. Last year, 1.5 million people, and it's done so for centuries. And in fact, this estimate published in 2013 cited the number 1 billion for the number of tuberculosis deaths. It's been around for a very long time, and the rate at which we are managing to control this disease is just not sufficient. So we need better interventions for TB, and there are many things that need to be done that include better treatments, better diagnoses. But of course, prevention is better than cure, and so preventive interventions are especially important. And of course, vaccination is prime amongst those. So here's the old dog. There is a vaccine for TB, and it's been around for almost 100 years. In fact, 99 years. BCG is a live bacterium. It was developed from a bacillus that infects cows and in fact causes TB in cows. It was attenuated until safe and was first administered to humans in 1921. So we've had this vaccine for a very long time. How is it that we still have this raging TB epidemic globally? Well, this is the problem. So this slide summarizes the efficacy of BCG at preventing pulmonary or lung TB. Lung TB is the form of TB that affects mostly older individuals, adults, and adolescents, and importantly is transmitted by those individuals to others, thereby maintaining the epidemic. And so what this slide shows is in each dot is a different study, an efficacy estimate from a trial. And you can see that in some of those trials, the dots are very high up, suggesting efficacy against pulmonary TB. But in some of these studies, those dots become very low and in fact even go below the line. The error bars denote 95% confidence intervals. And so wherever that confidence interval crosses 0, the efficacy is not significant. And so the take-home message from this slide is that efficacy afforded by BCG against lung TB is highly variable and often insufficient. So why do we even give this vaccine if it doesn't work well? And in fact, it doesn't really work well in most of the countries where the epidemic is raging. Well, the reason is this, that BCG also protects much more consistently against severe or disseminated forms of the disease in young children and infants. And so you can see on this slide, there were a number of studies that have shown this, and the dots are much more consistently in the upper regions of the efficacy scale. And in fact, this is so efficacious that we know that roughly or more than 50,000 lives are saved every year from BCG because it prevents disseminated forms. And we actually had a recent reminder of how important this is. In South Africa, this is a slide that shows the number of admissions for tuberculous meningitis or tuberculomas in young children at Tygerberg Hospital, which is not far from where we work. And so the bars denote the number of admissions every year, starting from 2008 to 2017, where you can see there's a massive spike. And this is because, or we believe it's because there was an international shortage of BCG vaccine. We certainly know that in South Africa, we were not able to vaccinate everyone, and so vaccination coverage dropped dramatically. And this increase was in the face of an overall diminishing TB epidemic, which is denoted by the blue line, which shows the overall case notification rate of all types of TB per year. So clearly a massive issue, and clearly BCG is not going to go away. It's unethical not to provide it because it saves lives. So then what does that mean for vaccination efforts? Well, I think we have to, of course, start with our understanding of how does this even work. So we do know that the first cell that is encountered when we breathe the bacterium into our lungs, the bacterium here is denoted as the little red bacillus, the little red bar. We know that alveolar macrophages are the first cells that encounter this bug. And these cells phagocytose the organism, which then goes into the phagosome, which is this little bubble that says kill. And usually macrophages are very good at dealing with organisms that they phagocytose. They can deliver through lysosomes some enzymes that are used to break down the organism. The compartment can be acidified, so the pH drops. And the macrophage can use activities like superoxide burst, as well as antimicrobial factors, like cathelicidin, for example, to kill these organisms and to eradicate that infection. The macrophage can also recruit other cells to the site of infection to help it out. And so that is what we call the inflammatory response. And we know that inflammation, for example, is a key thing in COVID disease, as it is in TB. So what is problematic with this picture is that Mycobacterium tuberculosis has evolved for a very long time with humans and has developed a whole string of excellent escape mechanisms, so that these mechanisms do not function very well and successfully. And in fact, the mycobacterium can live quite nicely inside the phagosome. It can replicate. It can even get out of the phagosome into the cytosol. It can replicate there and eventually spread to other cells and establish a very long chronic infection in the human that in many individuals leads to TB disease, tuberculosis disease. Of course, humans have not been sitting idly by. We've also evolved in the face of TB. And so one of the mechanisms that we can use to get around this is for the macrophage to call for help. And so that's what's depicted in the slide here. The yellow ball is another type of immune cell, where the blue is the macrophage that's infected. And so this type of immune cell is called a T cell. And so what T cells can do is recognize through a special receptor called the T cell receptor, tiny parts or protein fragments of the Mycobacterium bacillus that are presented through the MHC complex to the T cell. And when this happens, the T cell can be activated. It can produce molecules such as interferon gamma, as shown here. And this can then activate the macrophage to such a degree that it can engage additional processes to try and get on top of this bacillus and to try and kill it. So how do we know this, and what evidence is there for this type of mechanism? Well, a way in which we can nicely summarize this is actually from some early work done by a number of people. This slide was actually published this picture was actually published in 2004 by Robert North. And it depicts or summarizes a lot of experiments done with mice that are experimentally infected with tuberculosis. And so what's on the y-axis is the amount of Mycobacterium tuberculosis in the lung, and what's on the x-axis is time since infection. And so if you look at the line, it essentially exponentially increases for the first 3 weeks. And what happens after 3 weeks is that the adaptive immune system, those T cell responses have kicked in, and then they seem to be able to control the bacterial replication, not to the degree that the bug is actually effectively cleared from the mouse, but that it doesn't allow the bug to continue to increase its replication exponentially, but in fact it flattens out and stays at that stage for roughly a year. This is quite different to what we think happens in humans, but it's still a useful model to look at which components of the immune system seem to be essential. And so the WT represents a wild-type mouse where everything is functional. If you see the plus gamma delta, these are actually mice that are missing a special type of T cell called a gamma delta T cell. And you can see that when those cells are removed or knocked out from the mouse, nothing really happens. The mice are still exactly the same as the wild-type mice at controlling the bacterial infection. However, when MHC class 1, which presents peptides to CD8 T cells, is knocked out, such that CD8 T cells are no longer viable in these mice, then you can see there's a little bit higher bacterial growth in those mice, suggesting that CD8 T cells may play a role in controlling the bacterial infection. When MHC class 2, which presents peptides to CD4 T cells, is knocked out, you can see that there's even more loss of control of bacterial replication. The same is true for mice that lack alpha-beta T cells, such as CD4 and CD8 T cells. And then you can see that interferon gamma knockout and NOS2 knockout are the most, or allow the bacterium to essentially grow unabated, completely unabated. And in fact, those mice die very rapidly. So clearly interferon gamma, which is part of this Th1 pathway, T helper 1 pathway that CD4 T cells mediate, is critical for effective control in mice. What about humans? Well, we cannot knock out genes in humans, but there's the unfortunate natural experiment that the HIV epidemic has caused. And so we can see that in individuals who are infected with HIV, that the incidence of TB, which is depicted here on the y-axis, increases dramatically as people become more immunocompromised, as shown by declining CD4 cell count on the x-axis. And when individuals are treated with effective antiretroviral therapy such that the CD4 count recovers, you can see that the TB incidence rate recovers as well. It goes down as people have higher CD4 counts. So this is not definitive evidence because, of course, HIV infection affects a number of different immune functions, but it certainly does deplete CD4 T cells, and this can be used as one of the pieces of evidence that suggest that CD4 T cells are important in this mechanism of protection against TB. So we know a lot about protection from TB from BCG, and we know that BCG induces very good CD4 T cell responses, and that these T cells make interferon gamma very effectively. So this exact pathway, this Th1 pathway, CD4 cells that make interferon gamma, are indeed induced by BCG. But BCG only protects young children consistently. It doesn't protect consistently against pulmonary TB in adolescents and adults. So there are many theories for why this is the case. And one of the primary ones that was used in the early vaccine development was this idea here advanced by the late Ian Orme, that the primary flaw of BCG is its inability to effectively establish a population of central memory T cells. In other words, BCG induces this Th1 response, but it doesn't stick around for long enough. The memory function of those cells is not sufficiently long enough to protect the slightly older individuals that then develop TB disease. And so this formed the basis for a number of early TB vaccine products that were developed in the 80s and 90s as part of the new generation of vaccine development. And the first product that made it to clinical trials in humans was MVA-85A. This was a pox virus that expresses a modified vaccinia virus ankara that expresses a single protein that is made by TB and is also made by BCG, an environmental mycobacteria that we encounter in our environment. It's an enzyme, and it's highly recognized in humans who are infected with the bacillus, with TB. So the idea here was, because this vaccine was known to be quite safe, and because it's quite immunogenic, expressing this one protein and delivering that into human may then boost the immune response that's already there from BCG vaccination, such that we have better memory CD4 T cells, which can then protect against TB. And so here I'm showing some immunology data from a clinical trial that we performed here in South Africa. This was in infants who received BCG at birth, as is routine, but then who received either the MVA vaccine or placebo at about 6 months of age. And the data I'm showing here tracks the immune response, the T cell response, in fact, the number of T cells that produce interferon gamma on the y-axis at day 0, i.e. the day of vaccination, and then at various days after that. And you can see on the left that this vaccine induces a very nice boost in this immune response. It rapidly contracts, but that is what we would expect because the vaccine doesn't stick around for very long. But importantly, 6 months later, at day 168, we still see much higher T cell responses than they were beforehand. And you can see on the right that the placebo recipients had very low levels throughout. In fact, when we followed these children up for 3 to 5 years later, we still saw very high levels or higher levels of these T cells, suggesting that perhaps we've achieved or overcome this issue with a central memory T cell requirement. This then moved into a phase 2B trial, a proof of concept efficacy trial. Unfortunately, there wasn't the resources to perform a phase 3 trial were not available for TB for this vaccine. And so this was the next best thing. It was a trial that was conducted at Worcester, where we have a site, and we enrolled 2,800 infants half of whom received BCG, placebo, and half received MVA-85A. They were all vaccinated with BCG at birth, as is routine. And you can see on this slide that, again, we get this induction of the Th1 response, very nice levels of interferon gamma producing T cells. And then on the right, we even looked at other functions that these T cells may make. And so what we can see in the first bar graph from the left is that most of those cells that were induced were highly polyfunctional. They made interferon gamma, they made TNF-alpha and IL-2. So these are the type of cells that we hypothesized would be needed to protect an infant against TB once they became exposed to the bacillus. And so these infants were followed up for 2 to 3 years, and this was the result. So there were 39 primary endpoint cases, TB cases, so infants who developed TB disease in the placebo arm, and 32 cases in the MVA, the vaccine arm. And that translated to a vaccine efficacy of 17.3%, which was not significant. A very disappointing result. And one that made a lot of people sit up and think, ask questions. I just want to point out the endpoint 3 result here. In these trials, we often have a number of different endpoints because diagnosing TB, especially in young children, is extremely difficult. Endpoint 3 was essentially any infant or child that was put on TB treatment by their physician. And so you should be surprised by this number. 13% or 14% of trial participants ended up on TB treatment, which is an enormous number and illustrates the tuberculosis problem that we have here where we live. Nevertheless, of course, the result was very disappointing. It was the first vaccine to reach this stage of development in recent times, and vaccine hopes were certainly dashed. And in fact, it led to a lot of confusion, a lot of asking, well, what should we be doing? Where should our efforts be leading? A lot of funders thought about, well, is this really worth pursuing? And I think one of the things that we've learned, which I think we are learning again during this COVID epidemic, is that the simple answers are not always the correct one. But in fact, sometimes we just have to acknowledge that things are a bit more complex. Nevertheless, there were a number of other vaccine candidates that were in development. And one of the main things that people thought about is whether infants are in fact the correct target for these type of vaccines, given the lack of resources in the field. And so here's a modeling study that clearly illustrates on the y-axis, we have the number of cases per year of TB, that if we had a vaccine that could protect older individuals, such as adolescents and adults, we would have a much quicker impact. And in fact, of course, because these are the individuals that transmit the bug, we would protect the infants as well. And so we should focus on that population. So one of the products that was in development at that time already was a polyprotein vaccine comprising 2 proteins, shown here as MTB 32 and 39. This is developed or was developed by GSK and was formulated in an adjuvant AS01E, which comprises a TLR4 agonist called MPL, as well as a saponin derived from the soap bark tree called QS21, which was then put together in these liposomes and delivered into humans by intramuscular injection. And so as part of the quest to kind of answer whether this particular vaccine would protect against TB, we, along with many others, set out in a large proof of concept trial in quantiferon positive adults. So these were adults who already had evidence of being infected with TB. Roughly 3,500 were enrolled into this trial. And the trial was designed to ask whether vaccination of these individuals who were already infected could prevent them from developing active TB disease. And so they were randomized to receive 2 shots of this vaccine or placebo and were followed up for 36 months. And the endpoint was TB disease, slightly easier to diagnose in adults than in infants. But before I tell you the answer, I thought I would quickly show you what does this vaccine actually do in terms of the immune response. And so on the left, we have the profile of T cells that are induced in 2 different groups. These are actually adolescents, and this was done at a trial that we again conducted at our site. And you can see that in quantiferon positive individuals, so individuals who are already infected with the bacterium, we see a very nice boost at day 7 after the first vaccination. The vaccine was given twice. And after the second boost, this goes up again and actually reaches very high levels in some individuals. These are some of the highest levels of CD4 T cells expressing Th1 cytokines that I've seen. And these responses stuck around very nicely. By 210 days, it's still very much a high response. And even in quantiferon negative individuals who appear to be not infected with TB, after 2 shots, we see a very nice induction of CD4 T cells. On the right-hand side, we have an antibody response, and you can see after 2 shots, whether the people are infected or not, we see this very nice induction of anti-protein antibodies. So clearly, an immunogenic vaccine that induces very good T cell responses, would this then be sufficient to prevent TB? And here's the result. This was published last year. It's very recent and an extremely exciting result for the field. So what's shown here is a Kaplan-Meier curve. As these lines drop down, they denote cases of TB that have occurred. And you can see that the blue line is lower than the orange line, which means that the M72 vaccine was able to protect against development of TB, pulmonary TB. And in fact, it had a level of 50% efficacy. So very important result. So now comes the question, well, does that mean that these T cells protected against TB? Because if that's the case, then maybe we can use that information to make vaccines even better than 50% efficacious. And so the answer to that is we just don't know yet. But what we do know is that there have been 4 very, very well performed and elegant studies in non-human primates that were vaccinated with BCG and then challenged later on with TB. And of course, in a primate, it's much easier to dissect that animal once it's been killed, to look at what do the immune responses look like in the lung, how many bacteria are there, and exactly what correlates with protection. And so what we can learn from this is that T cells that produce these Th1 cytokines that I've been speaking about, plus IL-17, another interesting cytokine, as well as IgA antibodies, appear to be responsible for protection against tuberculosis. This is not definitive. It does not mean that this is exactly what's happening in the human, but we have some really concrete hypotheses that we can now take forward. And in samples collected from this M72 trial, we can go in with a hypothesis and ask whether these exact immune responses correlate with protection. So very exciting times in the TB vaccine field. And so I just have a few take-home points to finish off. The burden of TB is massive and requires warp speed intervention, much like we are seeing for COVID-19. It's been killing people for much longer and has been neglected for too long. Vaccines are necessary to solve this problem. BCG protection against lung TB is variable, but we cannot get rid of BCG at this stage because it protects young children. The first TB vaccine candidate that made it to efficacy trials failed, but we have success now in the TB vaccine field. And so the work really starts here because we need to now do phase 3 studies. There are other vaccines that are coming up in the pipeline that look also quite promising. And we need to figure out what is it that protects humans, because then we can work on getting higher than 50% efficacy. And with that, I will end and just acknowledge many, many people. This work is not our own only. This is always a very collaborative effort. In fact, it's a global effort. And there are many people at ERAS, at the University of Oxford, at GSK, at the Statham Serum Institute, at Sanofi Pasteur, as well as, of course, at SATVI, where I work, which is led by Mark Hathrell and has an incredible team of people shown in the picture below. Really fun to work with these people who are very committed to preventing TB. And of course, I also need to thank our funders, which are listed there on the right-hand bottom. Happy to take any talks, and I hope I didn't take too long.
No, you definitely didn't take too long. That was a really interesting story. I think that comparing TB to COVID-19 really puts it into perspective for a lot of us. So the audience, feel free to write your questions in the Q&A box, but I'm going to start with a question that I have, and it is, if the theory is that the BCG vaccine doesn't trigger sufficient long-term memory T cells, has there been thought given to booster shots every 10 or 15 years, where as needed?
Yeah, an excellent question. So they've in fact, many years ago, there were very large trials conducted to test this. And in fact, some countries used to give several booster shots of BCG to children. 2 large studies have been done, and both showed initially that there was no additional efficacy when boosted. However, there are some nuances in those trials, and so when one really kind of dissects that, it does look like that in individuals who have no prior immune sensitization to mycobacteria, there may have been an indication that there was some additional efficacy when BCG was given. So we have this major problem in the field where environmental mycobacteria and perhaps even TB itself infects individuals and induces a kind of immune response that seems to either block or mask the efficacy of BCG when it's given again. And in fact, that may also contribute to even a primary vaccination at an older age not being as efficacious as it is in young infants. Having said that, there has been a recent trial which was actually conducted here in Cape Town as well, which I couldn't speak about because of time, where we actually revaccinated adolescents that are definitely not infected with TB with BCG. And we saw a very interesting result there where it looked like it was able to prevent establishment of a sustained MTB infection. It did not prevent initial infection, but what happened is that people became infected and then seemed to clear the pathogen perhaps. It's not very easy to measure that because we can't measure the bug directly, but we can infer this from immunological tests which indicate this. So it looks like it's not going to be a great strategy because preventing TB disease with revaccination has just not yet borne out. It doesn't look like that's going to be a slam dunk, but perhaps there is something to it. And perhaps in future, one of the strategies that we will have is revaccination in some perhaps younger individuals with BCG, and then something like this kind of booster vaccine that I spoke about to prevent TB.
Great, thanks. We all have a question from Jag Kanwal. Jag, are you there? Would you like to ask your question?
I can read the question actually. Can TB remain latent in the lungs or elsewhere in the body and be triggered to multiply and invade cells after chronic stress? The answer is yes. So TB becomes latent. It infects cells chronically, and it can sit in the lungs in granulomas, which are kind of walled off cellular bunches. It can also go to lymphoid organs and other sites in the body where it can survive for a very long time. And in fact, does ultimately, if it leads to disease, somehow reactivates. And we don't know the mechanism by which that happens. But we know that there are a number of risk factors, and stress is one of them. The biggest risk factor is HIV infection, so immunocompromise of many sorts, and there are others like diabetes, etc. So that is exactly correct.
So let me ask you a question about the vaccine antigens, and it's similar to one of the questions that one of the audience members have. The BCG vaccine antigen and then also the M72 antigens, I'm sure this could be a talk in itself, but how were those selected? What is it about them that makes them ideal antigens?
Yeah, so BCG is the entire organism. It's a mycobacterium. It's got about 4,000 proteins. So it's a very complex molecule, organism. And it's actually not entirely known which of those proteins, and there are of course lipids and carbohydrates as part of that. And it's believed that perhaps all of those somehow work together to induce this kind of protective immune response. We don't actually know, so no one's really done the careful work to figure out which of the proteins inside BCG are responsible for protective immunity or not. M72, so those 2 proteins were chosen many years ago, partly by simply selecting antigens that seem to be immunogenic in humans who are infected with TB. So in other words, humans make an immune response, a T cell response to those proteins. during natural infection. But then also there were screens where these proteins were then taken into mouse models and that were vaccinated, and then they looked at protection. So, I mean, that might sound very straightforward, but in fact, it's not that straightforward, because there have been many different vaccine products that were developed that way and that did not show success. And so the mouse model is not an ideal model for TB. The mice show a very different course of infection. The immunopathogenesis is quite different. The immune system is quite different. And of course, they're not natural hosts for TB, etc. So I think that in terms of the antigens in that vaccine, we probably got a little bit lucky. It was an empirical approach and it worked, but we will take it and move forward and hopefully be able to improve on that.
Great, thanks. We have a question from Michael Reich, and I'm going to see if, Michael, would you like to join us and ask your question?
Sure, yes. Hi, Tom.
Hi, Michael.
I just have a question with regard to that vaccine trial you spoke about just a short while ago, where it indicated a 50% level of protection. Now, if you translate that into the real world, what kind of effect would that have on disease burden?
Yeah, that's an excellent question. At the moment, we can only go on epidemiological models that have been done. And certainly a 50% protective effect would already make a significant dent in the TB epidemic in terms of transmission. Of course, remember that in a model, we don't actually know to what degree transmission will be interrupted by this vaccine. But certainly in cost effectiveness modeling, we are 50% is worth it. I mean, it's worth developing that vaccine. We would ideally like to have something that's at least 60% efficacious, but at this point, we will take this. There will be a phase 3 study that will have much larger sample size and probably give us a better idea of the true level of efficacy. The confidence intervals were quite large in this trial, so the confidence intervals around that 50% efficacy. So I think we need to be a little bit careful about interpreting that to mean exactly 50%. It's somewhere around that point. We don't know how long it will last, the protective effect, which is really key when you're looking at a vaccine, because of course you cannot vaccinate people every 3 years. You want to have a vaccine that's perhaps at least 10 years or perhaps 20 years protective. And so those are things that we just don't know yet. So I would say the answer to your question is we actually don't know, but 50% is sufficient to kind of invest in this, to do that phase 3 study, and hopefully to try and improve on that with newer technologies and candidates and tweaks that may work to make that even better.
Great, thanks, Tom. I think in the interest of time, we should move on to our second speaker. So now I'm going to turn it over to Chaleka Mpande.